Hi everyone, can you hear me? I'm Nesrin Horzum. Okay, I'm Nesrin Horzum from İzmir Katip University. Welcome to the Hall 3 and Session 6. And before the presentation, I would like to remind you the time for the presentation is totally 15 minutes, including the questions or comments. So please try to finish your work in time. So the first speaker is Gamze Başbülbül or Ruth Moseko Piri, and the topic is determination of cyanobacteria diversity from hot springs around Aydın province by culture independent methods. Is the first speaker is here? No?
She seems not here.
The first speaker, I think, is not with us. So the second speaker is second speaker is here. First of all, I would like to ask. The second topic is here. There are many others in the second presentation. And then I'm moving the third one. The third one, I think, with us, Ibrahim. I'm seeing Yusuf Ibrahim. Is it the third presentation, Yusuf? Anyone hear me? Who is ready for the presentation of this session?
Hocam, the fifth presenter, he seems.
Which one?
Online. The fifth one. Zulaiha.
The third one.
Yes, like a BC 2004.
I think he or she is waiting for his turn, I think. The time is for the other slots, I guess.
Hello?
Hi.
Hi. Who is the speaker? Okay.
Hello.
Hello. Can you introduce yourself?
My name
couldn't see it.
Okay, I'm Zulaiha Gidado Muktar. Well, we have several papers together with other authors to present. I think is the fourth, or is the sixth paper, antioxidant capacity of 5 varieties of eggplants.
Okay, you can start your presentation.
Okay, thank you.
You can share your screen.
I don't know if you can see my screen.
Yes, we are seeing.
Okay. As I said, my name is Zulaiha Gidado Muktar, and on behalf of my colleagues, I want to thank you all for the opportunity. So I'm here to present on the antioxidant capacity of 5 varieties of eggplant fruits. So let's see. Sorry. Okay, the next slide. So the normal cellular activities that take place within living system, that is the sum of all the biochemical reactions and physiological activities, and several external factors generate free radical species. So whenever we have an imbalance between their accumulation and our body's ability to neutralize them, this leads to oxidative stress, which over time causes oxidative damage. This oxidative damage has been linked to aging and the pathophysiology of several chronic metabolic disorders, aging, metabolic disorders like diabetes, cancer, and Alzheimer's disease. So plants like eggplants, which is one of the most adaptable, low-maintenance vegetable plants in the world, it's also phenotypically highly diverse. We have a picture here depicting how diverse it is in terms of the physical color, its size, and its shape. So they are consumed raw and cooked by many cultures and used traditionally for a range of ailments such as hemorrhoids, heartburn, and skin disorders. And they have also been reported to be rich in antioxidant compounds such as phenolic compounds. And these phenolic compounds are known to have a positive impact in scavenging free radicals. So, recent studies, as I said earlier, have ascertained the relationship between the proliferation of oxidative stress and the contemporary dietary evolution, lifestyle changes, and the proliferation of aging, metabolic disorders, and poverty. So globally, this vicious cycle has had a negative influence on all facets of development. Eggplants' relative availability, affordability, their versatility, and their diversity makes them ideal for dietary inclusion and breeding programs, especially for poor rural households, strife-ridden regions, coupled with the looming threat of climate change. So this study will provide essential information for consumers, academics, nutritionists, breeders, and policymakers who are increasingly concerned about the health, functional, and economic benefits of crops and food they consume. So the present study has aimed to assess pH acid, then compare the total phenolic and flavonoid content and the antioxidant activity of the 5 varieties. So going to the next slide, which shows the methodology. This is just a summary, but we sampled fresh ripe eggplant fruits from Elazığ here in Turkey and Kano in Nigeria. So we used 5 different types which were categorized based on their physical appearance and local common names. So in the next slide, I have a picture of what they look like. We have the dark eggplant and the light purple eggplant, which were samples from. Then the next one is the bitter apple variety. Then we have the bitter tomato variety. And then the white garden egg. So the samples were extracted using methanol according to described methods. Then determination of the total phenolic and flavonoid content were also done using standardized methods described. I have the references there. Then we also then determined the free radical scavenging activity using ABTS and DPPH free radical scavenging assay. So the next slide shows the results, where the mean of the antioxidant activities determined and the total phenolic and flavonoid content of the 5 varieties are shown. So I have a bar chart which shows a clearer picture. The first one is for the ABTS, then the DPPH, the flavonoid and phenolic compounds. So for the ABTS radical, you can see that it has the free radical scavenging activity was higher than for the DPPH, while the total phenolic compounds were higher in concentration than the total flavonoids. So in the next slide, there's a bar chart on the side describing the comparison between the total phenolic compounds and the total flavonoid compounds. So all the varieties we tested had detectable levels of the phenolic compounds that ranged from 79.4 to 130 milligram, which was expressed as gallic acid equivalent of the extract value, which depicted a 1.64 difference in the content. The Turkish dark eggplant variety had the highest concentration of total phenolic compounds, while the Nigerian white garden egg had the lowest. In terms of total flavonoid content, it was found to range from 22 to 30 milligram. Sorry, it's a mistake here, of quercetin extract value, which showed a 1.4 of the total phenolic compounds was determined also in the dark eggplant, and the lowest was determined in the white garden egg variety. So across the different varieties, we are able to determine that a higher concentration and wider significant difference was observed for the total phenolic compounds than the total flavonoids. So based on our findings, we can say the Turkish dark eggplant, which has a deeper color, that is, it was the darkest in the picture than all the varieties sampled, and it also it had the highest content of both total phenolic and flavonoids compounds, while in contrast, the white garden example from Nigeria had the lightest color and also the lowest content of total phenolic and flavonoid compounds. So this is consistent with previous studies that have reported a similar correlation between phenotypic color intensity and total phenolic compounds of eggplants. So in the next slide is a comparison of the scavenging activity of the extracts from the 5 varieties. For the ABTS free radical scavenging assay, we determined it to range from 49.7 to 80.3 milligram per liter. For the DPPH assay, it ranged from 11.03 to 15.31. The extract had the highest free radical scavenging activity for the ABTS assay, while in contrast, it had the lowest for the DPPH assay. Then the bitter tomato extract had the lowest for the ABTS assay, and the white garden egg had the highest for the DPPH free radical assay. So, like I said, the dark eggplant exhibited highest antioxidant capacity in terms of the ABTS assay, while the white garden egg had the highest for the DPPH radical assay. In contrast, the lowest antioxidant activity was detected for bitter tomato in the ABTS assay, whereas the dark eggplant showed the lowest for the DPPH assay. So this variation in the antioxidant capacities of the different extracts is reflective of their various capacities based on their profile of phenolic compounds, and we can say other antioxidant compounds that they may contain to quench the ABTS and DPPH radicals in vitro. So correlating the concentration of the total phenolic compounds to the antioxidant capacity, it was observed that the dark eggplant variety sourced from Turkey had the highest concentration of total phenolic compounds and ABTS scavenging activity. So this correlates with previous findings that I have the reference here. So in conclusion, we can say that all the eggplant varieties we sampled for the study had an appreciable amount of phenolic and flavonoid compounds with varying levels of antioxidant capacities, and both the total phenolic and flavonoid content of the different eggplants showed disparities that are due to the varietal differences, growth conditions, and geographical origins of the samples used in the study. So these factors have been previously reported to influence their concentration in vegetables and fruits. So they may thus serve as cheap and available sources of antioxidants for dietary inclusion and breeding programs. So my next slide is the references. So thank you very much for listening.
Okay, thank you for the presentation. And any other friends, do you like to question to the presenter? Any questions? Again? No one? Let me ask one question.
Okay.
You conclude antioxidant activity of eggplants.
Yes.
You are especially making the antioxidant activity. Did you perform any other characterization of the eggplants? It is, I mean, the content of the eggplant or nutritions inside the eggplants.
Well, we did literature review and then we assayed other vitamins and amino acids.
Okay, thank you.
Thank you.
Are you a PhD student or master student?
A PhD student.
In Turkey or?
Yeah, in Turkey. I'm presently here.
Okay, nice to meet you.
Same here.
I think the numbers are changed, I think. Now we will turn back to second presentation, I think, the BI 2007. The topic is indices of the oxidative stress in experimental rats exposed to smoke of the some compound-based mosquito coil insecticides. This is your turn.
Good evening, everyone. I hope you can hear me.
Yes, now we can hear you.
Okay. So my name is Mohammed Salihu Ibrahim. I am from Nigeria, and I'm a PhD student in Varieties University in Turkey. So today I'm going to present our research titled Indices of Oxidative Stress in Experimental Rats Exposed to Great Dosages of Meperfluthrin-based mosquito coil insecticide. Due to time factor, I'm not going to waste much of your time, so I will just quickly try to go through malaria, because this mosquito coil insecticide is being used against mosquito in especially West Africa region. And the reason, we are having so many cases of malaria. So malaria, as you can see, is a life-threatening disease, which is caused by the parasite Plasmodium carried by a mosquito. So these insecticides that people are using consist of this, what we call pyrethroids. And these pyrethroids are different types. We have transfluthrin, we have meperfluthrin, and we have D-transalatrin. So in this work, we decided to make use of the insecticide that is being used against these mosquitoes, which is Goldia mosquito coil, and it has this meperfluthrin as its active ingredient. The reason why we decided to conduct this experiment is because, people in our region are in the habit of sleeping with this insecticide. So we don't know whether this smoke will have any health problem. So that is why we decided to conduct this experiment. And as you can see, our country, like Nigeria, is suffering or having the highest incidence of malaria globally. And according to the World Health Organization World Malaria Report that was published in 2009, it has been said that 25% of the whole malaria death globally is in Nigeria. So without wasting much of your time, let's go quickly to the experimental design. Okay, so we divided, we get experimental rats, and we divided them into 5 groups. We have group G1, G2, G3, G4, and G5. Each group consists of 4 rats. Group G1 were exposed to 15 minutes of this insecticide. Group G2 were exposed for 30 minutes. Group G3 were exposed to for 1 hour, and group G4 for 2 hours. Group G5 served as control. They were not exposed. They were kept in a separate cage. During the exposure time, we transfer them to the cage where we carry out the exposure. After exposure, we transfer them to the cage where we keep them. This experiment was conducted within the period of 4 weeks. So on the 29th day, the rats were sacrificed and blood samples were collected, and the blood samples were centrifuged and the serum was separated and used for biochemical assay. So this is the result. We decided to check, there is markers of oxidative stress, malondialdehyde, and then we conducted a dry layer oxidative stress test because we want to see whether there is evidence of oxidative stress in the blood of these experimental animals. And then we include serum bilirubin and serum alkaline phosphatase for the integrity of the liver cells hepatobiliary system. So when we compare the result of bilirubin and alkaline phosphatase with control, there is no significant difference from group G1 to group G4. When we compare them with control, there is no significant difference. But for serum malondialdehyde, comparing group G1, G2, G3, and G4 to group G5, there is a significant free which gives rise to high production of malondialdehyde. So oxidative stress, increase in the dry layer oxidative stress. So this is the result for malondialdehyde. As you can see, this is the control. So when we compare G1, G2, G3, and G4 with control, there is a significant increase, as you can see. And the next slide is showing the dry layer oxidative stress, because this experiment was carried out on the whole blood taken from the experimental rats. This is A, B, C, and D. A is a member of group G1, B is a member of group G2, C is a member of group G3, and D is a member of group G4. So usually when we have a normal coagulation, it is telling you that there is no stress in the blood. But when there is protein puddles and crack in the blood, is an indication of stress in the blood of these experimental animals or of an individual. So if you can see group C and D, there is crack and white protein puddles showing a disconnected or poor coagulation. So this is telling you that the members of group C and D, that is the members of group G3 and G4, suffer from more of the oxidative stress, which is attributed to the exposure time. And this is the control. If you can see, it is a uniform coagulation for the control when you compare other groups with the control. So the exposure of this mosquito coil results in significant increase in MDA level in the experimental rats compared with the control rats, as I said earlier. And this may be due to the production of these free radicals, because MDA is a reactive aldehyde generator as one of the products of lipid peroxidation and the most widely used index of lipid peroxidation. And the increase in serum that leads to the production of more free radicals, which increases lipid peroxidation and eventually increase the generation of reactive aldehyde, the MDA. Because as I said, there is no significant difference when we compare bilirubin and alkaline phosphatase results with that of control. There is no significant difference. But for group G4, there was found to be a significant decrease in total bilirubin. So we can correlate this with, because bilirubin also have an antioxidative function, and because of increase in that's why there is a decrease in total bilirubin. The high stress is not a disease. Existing diseases. So many related to increase in production of free radicals. They complicate so many conditions. So that is why it is very, very important to look at it. And further experiment is going to be carried out. We found some volunteers down in my country, and they are ready to give us their blood sample, and we are going to include hydroxyneonal also, and also the level of some antioxidants like vitamin E. Thank you so much for listening. If you have any question, please do not hesitate to ask. Thank you.
Thanks for the presentation. Any questions to the speaker? No one? This is not a question, just advice. I'm suggesting you try to keep your slides simple and basic, and avoid using the whole sentences in the slides, because it is very difficult to follow to the listener. Just kind of advice to you.
Yes, thank you so much. because I prepare it within a very short period of time. That is why a little unorganized. But thank you so much for the advice.
You're welcome.
Yes.
Good luck for your studies.
Yes, thank you so much.
I think our last speaker, and this is the title is Evaluation of some defense enzymes activities in chickpea plants under cold stress. I hope we will listen the last speaker. Yes, finally.
Hi.
Hi.
Good afternoon.
Good afternoon.
Dear attendees, thank you for having me. Can you hear me?
Excuse me, Mohammed Ibrahim, could you please stop sharing your screen?
Yes, okay. I'm just looking for, sorry.
Can I continue?
We are having a problem here. I want to stop my screen sharing. Okay. You may proceed.
Salih, you have to close your voice. This is the unmute.
Okay, now you can share your screen.
Can you?
Yes, we can hear you.
Can you hear me?
Yes, we can. Okay, we can hear you now. You have 15 minutes.
Okay. Hello, dear attendees. Thank you for having me. I'm Hutan. I'm a master's student at plant breeding at Ege University. And I'm going to present a study related to investigation some defense enzymes in chickpea under cold stress. And can you hear me well?
Yes, we can hear you. Continue, please.
Due to the immobility of the plants, they are always exposed to abiotic stress, such as cold stress, drought stress, and therefore they have to perceive the environmental changes, and they have to respond to these changes to maintain their homeostasis. And cold stress as a contributing factor for plant growth has many impacts on the quality and quantity of the plants, and physiological, molecular, and biochemical responses to cold stress originate from genome reprogramming and changing the activities of induced genes. Plants require different defensive mechanisms for being survived and tolerant to environmental stresses. And in the present study, the activity of defense enzymes in two genotypes of chickpea, which one of them is susceptible one and the other one is a tolerant one, under 4 degrees Celsius cold stress were evaluated. The enzymes which will be evaluated is alternative oxidase, or in abbreviation AOX, superoxide dismutase, or SOD, and catalase. The chickpea is an annual legume of the family Fabaceae, and there are many difficulties and barriers for spring cultivation. For example, drought and lack of moisture at the end of the season leads to a reduction in production and the product yield. And as a result, autumn cultivation.
Sorry. We just seen the title page of your presentation.
Oh,
Is
really?
there a problem or you passed the page?
What? I have changed the pages?
We are just seeing the title page. Can you try again?
Yeah, I try to. I don't know what's the problem.
Are you changing now?
I'm looking for a different way to show you, but I have changed the slides. I don't know why you don't see the pages.
You can try stop sharing your screen and share it again.
Okay. And can I join the meeting again? Maybe it will be solved.
Okay, you can try. Can you see? Yes, we are seeing your desktop now.
Yeah, I have opened the PowerPoint again.
Mhm. The previous one was not the PPT file. It was PDF file, maybe for this reason.
I have opened the PowerPoint again.
Mhm.
Okay, I think you can see it.
Perfect. Perfect. Uh-huh.
Okay. Okay. So, as I said, I was talking about Kicker argentinum. It is an annual legume of a family of Fabaceae, and there are many difficulties about spring cultivation, and as a result, autumn cultivation in regards to a high level of moisture and rainfall seems more reasonable. However, cold stress can be a limiting factor against plant development. And in conclusion, investigation of the defense mechanism of chickpea during autumn in the form of susceptible and tolerant genotypes can be very useful for breeding schemes. The first index that we have studied is electrolyte leakage index, and ELI, or electrolyte leakage index, plasma membrane is the first part of the cell that responds to the cold stress, and therefore measuring the amount of damages to the plasma membrane through the electrolyte leakage index in the early stages can be an appropriate factor for characterizing the durability of plants against cold stress. On the other hand, reactive oxygen species, or in abbreviation ROS, including superoxide ions, hydroxide radicals, and non-radical molecules such as hydrogen peroxide, are one of the major factors that contribute to cellular damages. ROS are one of the products of electron transport chain in the chloroplast, mitochondria, plasma membrane, or it can be as a secondary metabolite, and environmental stresses such as cold increase the amount of ROS due to the disruption in cell balance, or as we know, cell homeostasis. An excessive increase in ROS or their inefficient removal put the cell into oxidative stress. Effective removal of ROS under cold stress requires the activation of defense mechanisms. Antioxidative enzymes such as superoxide dismutase, catalase, and some other enzymes can be mentioned. The role of alternative oxidase is another enzyme that I'm going to talk about. The role of AOX in the electron transport chain in turning the oxygen into water. AOX is controlled by a gene family including two subfamilies named AOX1 and AOX2 in the cell nucleus. And the mentioned gene family are affected by biotic and abiotic stresses. And in addition of lack of proper operation of the electron transport chain affect the expression of these genes. AOX plays an important role in ROS reduction under cold stress so that the amount of ROS and consequent cell damage are reduced by rising the activity of this enzyme. And because I don't have ample time, I will skip this part. This is material and method. I just want to mention about some general points. The seeds were supplied by an institute in Iran. The seeds were disinfected and then germinated on filter paper, watered by distilled water. And then the seedlings transferred to the pots, and the pots were placed in a growth chamber. The temperature was 23 degrees Celsius, the moisture was 75%, and the growth time was 21 days. A number of samples remained at the mentioned condition as a control sample, and the parts transferred to 4 degrees Celsius condition, and they were kept for 6 days. Sampling was performed on the first, third, and 6th days. Electrical conductivity was measured by EC meter, ELI measured by this formule, AOX activity measured by a spectrophotometer, SOD activity measured by some methods, and hydrogen peroxide measured by a spectrophotometer, and we analyzed our data by SPSS 20 software. The results of the variance analysis demonstrated that the interaction effect of genotype and temperature treatments had meaningful differences in 1% in terms of the study traits, which indicated the genetic diversity among genotypes and also indicated the different reactions of genotypes under cold stress. In the present study, ELI is considered as a damage indicator, as you can see in this bar chart. And in general, the low rate of ELI indicates the high resistance and more active defense mechanisms of the plant under cold stress. ELI comparison means test represents a meaningful difference between two temperature treatments, and as a result, a significant difference can be expected between two genotypes. The ELI in the tolerant genotype under 23 degrees Celsius was remarkably less than the susceptible one, and it shows the distinctive genetic capacity of resistant or tolerant genotype. As you can see in the bar chart, the black column refers to the tolerant genotype and the gray column refers to the susceptible genotype, and on the first day under cold stress, transferred the seedlings to 4 degrees Celsius, didn't make any significant difference in ELI, and the ELI in the susceptible genotype had a remarkable rise on the 3rd and the 6th days under the cold stress. However, there was no big difference in the amount of ELI in the tolerant genotype in comparison with the control sample. And also, the highest and the lowest amount of ELI recorded on the 6th day for the susceptible and resistant genotype, respectively. And let's have a look at the bar chart of hydrogen peroxide. The culprit of a high rate of ELI under cold stress is reactive oxygen species, which leads to oxidative stress, and therefore the amount of hydrogen peroxide has been measured in the present study over the different days. Unlike other rows, hydrogen peroxide has a longer half-life and it's able to spread through biologic membranes. And hydrogen peroxide also is able to trigger oxidative stress as a messenger molecule in an area away from the produced site. The dried results represent that the amount of hydrogen peroxide in the tolerant genotype after a meaningful rise in the 1st and 3rd day of the experiment drops in the 6th day, so that under such a condition, it fell to the lowest level among experimental treatments. On the other hand, the amount of hydrogen peroxide in the susceptible genotype rose dramatically in comparison with the control sample. And it was by about 33%, as you can see in the figure 2. The present results confirmed the previous results from ELI measurements. Furthermore, it demonstrates the extension of oxidative stress in the susceptible genotype under cold stress. Superoxide dismutase was another enzyme that we investigated and is an active molecule in the defense mechanism of plant cell and plays a vital role in turning superoxide into ordinary molecular oxygen or hydrogen peroxide. The level of superoxide dismutase activity in the susceptible genotype after an inconsiderable change in the 1st day decreased in the 3rd and 6th days, so that the amount of superoxide dismutase reached the lowest level. But on the other hand, the level of SOD activity in the tolerant genotype increased gradually and reached a peak of 50% on the 6th day. And catalase is an active molecule in the defense mechanisms of plant cell and plays a crucial role in hydrogen peroxide degradation. On the 1st day, the activity of catalase in the tolerant genotype, as you can see, was reduced, but it rose significantly in the 3rd and 6th days and reached the level of catalase in the control sample. The activity of this enzyme in the susceptible genotype had a reduction of about 52% on the 3rd day compared with the control sample. Although the activity of catalase grew on the 6th day compared with the 3rd day, it had a remarkable drop in comparison with the control index. And finally, the alternative oxidase was our final enzymes that we evaluated. In the next steps of the study, the activity of AOX as an important enzyme complex in the mitochondrial electron transfer chain was investigated. Having taken longer time from the 1st day to the 6th day, the activity of AOX after a significant reduction indicated an upward trend, which represents an effective role of this enzyme in biochemical responses to cold stress. However, the rise of AOX activity in the tolerant genotype was more than susceptible one considerably. And as a result, we can say that the maximum activity of AOX was seen in the tolerant genotype on the 6th day of the cold stress, whereas under such a conditions, AOX activity in the susceptible genotype significantly was lower compared to the tolerant genotype. The increase in AOX activity, which was accompanied by a decrease in cellular damage, as we know, ELI, electric leakage index, and hydrogen peroxide results, particularly on the 6th day of cold stress, has indicated the significance of alternative oxidative enzyme activity in the resistance of Chickpea to cold stress. And it would seem that AOX biological pathway and the pathways of antioxidant responses, such as catalyze and superoxidase nucleotides, accompany each other to inhibit hydrogen peroxide and electrolyte leakage index further production. AOX increase under cold stress indicates that the ROS inhibition process may be regulated at the level of transcription or translation. Studying the activity and mechanism of these enzymes and the strategies to increase the activity of these enzymes in Kicker arietinum under cold stress in sand breedings makes the autumn cultivation possible. Thank you for your attention and your patience. I'm here to answer any questions that you have.
Any questions to our last speaker? No questions? No wondering anything?
I'm just curious.
Ask again.
Okay, I'm just curious. You're an agronomist and a biochemist. So I noticed you just concentrated on measuring enzyme activity relating to cold stress. For us, we normally measure others like malondialdehyde or 4-hydroxynonenal or glutathione.
I can't hear your voice well.
Oh, okay. Can you hear me now?
Yes, yes. If you can speak a little louder, I can.
Can you hear me?
Yes, yes.
Okay. I'm a biochemist. So normally when we're measuring oxidative stress or any type of stress, we concentrate on MDA or 4-hydroxynonenal. So do you also do that for seeds or is it only the enzymes?
I can't hear you well again. At the beginning, I can hear you, and then I can't. I don't.
Maybe you can chat. You can text to him.
I will send a message.
Yeah, please send a message. Thank you.
You can use the chat, group chat now, and
Okay.
he
I will
can
answer
see
you
your
on chat. I will answer you on chat.
Nesrin hocam.
Efendim hocam buyurun.
Kolay gelsin öncelikle. Arkadaşlar hepinize merhaba.
Teşekkürler.
Diğer 3 katılımcı görünmüyor herhalde.
Evet gelmediler. 3
Bir
arkadaşımız
önceki
konuşuyor.
oturumda session 3'te oraya biraz geciken bir arkadaşımız oldu. Müsaade ederseniz Can-Claude şimdi bağlandı. AE 2001 sunumunu yapabilir.
Bir soru cevap var
sonra,
da
evet.
ama hani vaktimiz var diye ben de müsaade ettim.
Daha var vakit zaten hocam.
Aynen.
3 kişi gelmemiş.
Sonra diğer arkadaşın başlığını bilmiyorum ama yaparız.
Ben buradan takdim ederim hocam.
Devam edelim.
Soru uzun galiba.
Gerçekten.
Hello. Can I start?
Jean-Claude.
Hello.
Yes. Just a few minutes wait for a Jean-Claude, just wait for a few minutes. Soru geldi herhalde.
Hı hı.
Can I quickly ask you a question, please?
No.
Thank you. Thank you for your presentation.
Mukhtar.
Thank you for everybody. Actually,
Hello.
about data. So if you present, if you present some
Hello.
data so
Hello.
you can see it, it would be better, I guess. That was like just a remark.
Hello.
Jean-Claude,
Sen
birkaç dakika sonra seni alacağız. Bir soru var. 2 dakika bekle, ben söz vereceğim sana. Tamam.
Utan, did you see the question?
Yeah. I have seen, and I'm typing.
Okay.
And also, I can answer by voice.
Yes, please.
Yes. This study has investigated some enzymes involved in defense mechanisms of this plant, Kicker arientinum. But there are some other parameters and factors that should be investigated, same as MDA or other enzymes. So these are just some enzymes involved in the defense mechanisms of Chickpea, especially in this species. So we should investigate other enzymes related to this matter.
Thank you very much.
Okay. Is it convincing? Okay. And also, I would like to ask, what is your future plan? You investigate the enzyme, different enzymes in the chickpeas under the cold stress. What is your future work?
What is my?
What is your future work?
We are going to investigate some other enzymes related to ethylene pathway as for a tomato. And we are going to investigate these enzymes and its pathway against another pathogen, canker.
Okay. Okay, good luck for your studies.
Thank you so much. Have a nice day ahead.
You too.
I'm curious to know which software did you use for that.
Sayın
I
hocam.
think we have... Buyurun hocam. Buyurun hocam.
Sorular bittiyse diğer arkadaşımıza uygunsa ben
Tabii,
bölmüş
tabii.
olmayayım ama.
Yok, estağfurullah.
Arkadaşlar, Jean-Claude Tuysengen'in Comparative Study of Composted Greville Robusta Paspalum and Anus Paspalum Biomasses başlıklı bir sunum var. Jean-Claude, you may start.
Benim kendimi göstersene.
Teşekkür ederim. Evet, şimdi Neurometod training yaparken bir program aracıyla yapılıyor. Biz hesaplayarak yapmıyoruz. Şimdi onun için datalar hani çıkartmamız
Jean-Claude, do you hear us?
bir program üzerinde yapmış olduğumuzu bilmek istiyorum aslında da. Hangi program kullandınız? O programda Levenberg, Marquardt algoritma mı kullandınız?
Jean-Claude duymuyor herhalde hocam.
Bu kavramı söyler misiniz?
Hocam ben bir daha arayayım telefonda da.
PV systems.
Hello.
Yes, you may start Jean-Claude. Jean-Claude başlayabilirsin. Söz sende. Seni dinliyoruz.
I'm facing a technical issue.
Shall we wait or later?
I'm coming. Yeah, I'm trying to open the document. I opened the document and it was closed immediately.
Okay. Hocam uzaktan da böyle biraz sıkıntıları oluyor. Technical problem diyor. Görüntü biraz geç geliyor galiba. Sunumu açmaya çalışıyormuş herhalde.
Can I start before opening my presentation? I don't know what's going on.
Yes, you may start, Jean-Claude.
My PowerPoint presentation is not being open.
If there is a problem, let's postpone your presentation to tomorrow.
Is that speaker? The problem is that I can't share my presentation.
Okay, then let's postpone it to tomorrow. Nesrin Hocam, buyurun söz sizde. Biz arkadaşa yarın bir fırsat daha verelim.
Tamam
Sanıyorum
hocam.
katılamayacak.
Görüşmek üzere. Aynen. Okay, this is the end of this session. Unfortunately, due to the COVID, we are doing all the meetings, conferences, lectures online. Hope to see you all face to face in healthy days. And good luck for your master and PhD studies. Have a nice day.
Thank you, everybody.
Okay.